ഞാൻ നമ്മുടെ ഇതിനകത്ത് എൻ്റെ പവർ പോയിന്റ് പ്രസന്റ് ആർക്കറിയാം ഇല്ല ശബ്ദമില്ല. അതിന്റെ ഇതെല്ലാം കിട്ടിയോ?
ഇല്ല, അതിന്റെ കൊട്ടേഷൻ സോഫ്റ്റ്വെയറിന്റെ ഇന്നലെ വന്നതേ ഉള്ളൂ.
Good evening, Dr. Daniel. People are still joining. What should we do? Should we start or wait for few more minutes?
Yeah, you can wait for some time.
Okay. Thank you.
Hello, madam.
Yeah. Sir, we will start.
How much time I should take? 1 hour?
Up to you. You can take as much time you can.
No, if I get 1 hour, I can go slowly.
Yes,
So
you
that-
can get 1 hour, sir.
Otherwise, I have about 100 slides.
You can go take 1 hour, sir. No, not an issue. Should I start now? People are still joining, but anyway,
No,
they can.
you introduce me, that
Yeah.
takes some time.
Yeah.
Okay.
Okay. Good evening. On behalf of Department of Botany and RD National College, I extend a warm welcome to our guest, Dr. M. Daniel, all the faculty members, scientists from various institutions of the country, and my dear student participants from across the university colleges, and so on and so forth. We are fortunate to have a renowned personality, Dr. Daniel, amongst us today. Dr. Daniel is professor and ex-head of the department, botany, and former Dean of Science, MS University, Baroda. Presently, he is the managing director of Dr. Daniel's Lab, Baroda. He has a teaching experience of 39 years and research experience of 49 years, and he is still continuing with his research. Dr. Daniel has a long biodata, but I will be making it as short as possible so that we get more time to listen to him. He has published 170 papers and 18 books, both by international and national publishers, scientific, reference book, and popular books. He has 167 research articles, guided 17 PhD students, successfully completed 5 major research projects with funding of around 69 lakhs from the funding of University Grants Commission, NMPB, and other funding agencies of the country. He has holding several key positions, such as is a member of Research and Advisory and Monitoring Committee for Botanical Survey of India, appointed by MOEF, Government of India, member Advisory Board, Mentor Biological Science, Nagarjuna University, Baroda, member of Board of Studies in Botany, Nagpur University, and visiting professors in many universities. Dr. Daniel has been awarded Lifetime Achievement Award in Life Sciences by Indian Society of Solid State Chemists and Allied Branches in 2015, and Mahatma Manava Seva Award in 2015, the highest recognition from Syrian Christian Mahatma Church, Kerala. With this short introduction, I request Dr. Daniel to give his presentation amongst us. Welcome, Dr. Daniel, and thank you for accepting our invitation.
Thank you, madam, for the nice words that you have presented. Now, hopefully the screen is visible, right?
Yes, sir. Yes,
and
sir.
the sound is okay?
Yes, sir.
Now, my today's topic is very close to my heart. That is on chemotaxonomy and bioprospecting. And I am really happy that Professor Ajit Kumar sir from Bombay is there. He was asking me about research topics in this. And you got lot. That is why the subject of bioprospecting is very prospective. And chemotaxonomy is classifying plants based on their chemical characters. Just like the blood groups of members of the family, they have similar blood groups or certain DNA characters are the same. The plants also within a group, whether it is a genus, a species, varieties, or families, or even order, or even classes, they have certain chemical characters. But this was very poorly known before 1970s. Probably I started my research in 71. I did my MSc in Kerala and then came to Baroda for PhD. At that time, I had some idea about that. You can classify plants based on their chemical characters. So I told my guide, why can't we start them? He said that where is the literature, where is the expertise? Then I said, sir, we'll try to find out. We read a number of articles, probably from J.B. Harborne and all that. And, actually, the first book on chemical plant taxonomy came in 1963. And second book, 1 year later, Chemotaxonomy der Pflanzen, is a German book. by Hegner. That means it is German and you cannot read it. And then in 74, Darnley Gibbs, he brought out a voluminous book on chemotaxonomy. So these are the first books defining what is chemical plant taxonomy. And before that, people thought of doing something. But if you see the next slide, there were no methodology available. So in 1973, J.B. Harborne published the first very good book, Phytochemical Methods, which is still very useful. And then along with that, Mabry and Associates, they put another very good book on systematic identification of flavonoids. That also is in 1970. Now, there were a lot of technical advancements in the fields of paper and thin layer chromatography and spectrophotometry. So you could test plants for their chemical components in the lab itself. Otherwise, this was a prerogative of organic chemists. And they, their disadvantage, my son is an organic chemist. His disadvantage is that he needs large amount of plant material, maybe 50 kilos or 100 kilos, then he will isolate and get 1 or 2 grams or even milligrams. So with the advancement in paper and thin layer chromatography and column chromatography, now GC, LC, LCMS, every facility is available now. We can even identify the compound present even in a single herbarium, even in a single leaf. Now, before entering into what are the characters you can use in classification, we call them as chemical characters. And everybody knows that what are the compound present in plants. See, plants, their chemistry are entirely different from animals, though they have some common things like certain carbohydrates, proteins, lipids, which many of the biochemistry books are giving. But in biochemistry books, plant metabolites or plant chemistry is very poorly written. So you can classify the compound present in plants as one, primary metabolites. Primary metabolites are always being produced and used over. And there is a metabolic pool. Always it will be there. On one side, these compounds are being produced, and the other side, they are being used up. So there is a turning over of these compound cycle, production and utilization. So they form a metabolic pool, and they are almost same in all the plants. So they may not have much of a taxonomic value, but you can say that these are unifying characters. You can say the plants form one united group because of their primary metabolite. They are different from animals because of their certain primary metabolites. When we come to secondary metabolite, and at that time, when I started my work in 1970, many of you might not have been born also. At that time, I was a struggling boy of 21 years with no support and nobody who can guide me. And then this was in my all elders, physiologists said, what are you going to do with your secondary metabolites? Secondary metabolites are hopeless. One of my senior professors said, Daniel is working with dead compounds while he was doing tissue culture. So he said he was working with living compounds while I work on dead compounds. Now I am very happy that I was working on dead compounds. So physiologists or biochemists used to call them excretory products. The plants do not have an excretory system, so they store them in leaf, bark, and all that. Some people call them end products, and some people call them as byproduct. They are formed accidentally. And even Heslop Harrison at Taxonomy said they are luxury diversification. See, like a rich man will have lot of trophies and things in his drawing room, which are not actually useful, only to show off. So they are the luxury diversifications. So these compounds were termed by a taxonomist, Heslop Harrison, as luxury diversification. They were thought to be useless. But now, believe me, in 2021, after about 50 years of research, I say the survival of the whole plants depends on these secondary metabolites. Now, I'm giving a Facebook story of evolution of plants. Now, I have started only last week. There I am explaining the evolution in different groups of plants and how they have produced different metabolites which help them in struggle of existence and surviving. Now, they are not byproduct, they are not excreted product, they are produced by specialized metabolic pathways. Remember, they are produced by specialized secondary metabolic pathways. They are gene controlled and they are not metabolized at all. Even though at some cases you can see that there is a gradual turning over of this compound. They are the survival strategies of the plant. See, in my Facebook story, I said I was explaining about algae. Algae could not come to the land. At that time, the land was barren, no ozone layer. The algae cannot come to the land because of the ultraviolet rays. And it is the Cara, that amphibious algae which developed flavones as UV screens. So Cara is rich in flavonoid and that immediately taken over by bryophytes. Bryophytes elaborated large number of flavonoids. That is why they could withstand the hot sun. And then when you come to higher plant lignification, and then, at that time, a number of insects came by the time pteridophytes were there. And what of insects? There was volatile oil. Then there were herbivores. And what of them? The plant produced alkaloids and many other compounds. So it is these group of compounds which help the plant to survive. That is why the angiosperms became the largest taxa within 40 million years. And Darwin says that it is something of a wonder. He says that it is because of their secondary metabolites. And we come to secondary metabolites now. Now, why should you know about phytochemistry? Now people talk about, now more emphasis on your foods. Food as your medicine. That was our old slogan. And, food has to be even Aristotle said that. Food has to be your medicine. See, now they say nutraceuticals are there. You take flax seed, you take this seed, you take chia seeds. Why? They say that they contain something very good. But my dear friends, all these things are available in your common foods. You are wasting your money on those, imported ones. You just see all foodstuffs like cereals, pulses, vegetables, fruit, nuts. They contain secondary metabolites like phenols, polyphenols, non-protein amino acids. All spices, they contain volatile oil. They contain secondary metabolites. All beverages contain alkaloids, secondary metabolites like caffeine, alkaloid, and tannins. And all medicine, now they say that people will say you take Giloda, or you take Giloy, Tinospora, Amrita, you take this and you will be immune to everything. But they do not know what actually they contain. Now, we have developed a lot of phytochemical applications in MS University, in herbal cosmetics. Shaina Hussain, she was preparing herbal cosmetics without herbs. Now, natural perfumes, very body-friendly perfumes. You get gums and resins, all masticatories, fumitories, and narcotics, woods and silky chemicals, gums are unseen, natural colors, biopesticides, biofuel, natural dyes. All these are the branches of plant chemistry and chemotaxonomy. So therefore, we introduced, because I started work on the chemotaxonomy, the people, my boss and all that, they said that you do not go back to Kerala now. We will give you direct appointment. So we have revised all our syllabi on economic botany, and there was no book. So my first book came in 1990 along with my guide, S.D. Subnis. a phytochemical approach to economic botany. And that was a student edition, only rupees 65. Now, in chemotaxonomy, I started work in 72. So using chemical characters in classification and inferring phylogeny. Now, which characters are best? They should be stable. Means there should be enough time between isolation and identification. It should not decompose very easily. Then it should not be very difficult to identify them. Then that will discourage people from using them. So ease of identification is another thing. Then higher contrasting state. See, they should be having a high distribution. There should be plants with them. There should be plants without them. But that also is controversial if they are uniformly present. That is a phylogenetic character, like chlorophyll. See, all plants contain chlorophyll, so they form a natural group, while fungi does not contain chlorophyll, so it forms a different group. So even if higher contrasting states are not there, you can use them. And they should not be influenced by heat, weather, or any other external factor. These are the 4 characters for a good chemical character. Now, even though we say primary metabolites are not much useful as chemical characters, I will give you some examples. You know that if you are a diabetic patient, earlier, the fruits are, doctor used to say, never touch fruits. But now they say that you can take apple, or you can take any fruit of rosaceae, raspberries, blueberries, anything. All those things are there. There are many fruits in rosaceae because they do not contain glucose, they contain sorbitol. And sorbitol is directly converted to fructose 1,6-diphosphate. So there is no problem of metabolizing glucose. And you don't need insulin for that. Then bornacitol, that is another sugar alcohol in gentianaceae. I proposed a family hexaceae away from gentianaceae way back in 78 because they do not contain bornacitol. Then raffinose series, there is a group of di, tri, tetra saccharides found only in Scrophulariaceae. Then there are polysaccharides which are characteristic to certain groups. All legumes, all the seeds, they have a vitreous layer below the seed coat. There is galactomannan. This is one of the best fibers. And the non-protein amino acids are there. And many people do not know. If you ask them how many are the amino acids in plant, they say 25 or 26. I say it is 600. See, the protein amino acids are only 25 up to 30. But there are large amount of non-protein amino acids. They occur as free amino acids or peptides or in other combination. Half of these are in leguminosae. Leguminosae is great because they have that nitrogen fixing bacteria. So it produces so many non-protein amino acids as a form of nitrogen, maybe for defense or fight between plants, allelopathic interactions. Then fatty acids, they are very common, but see in Flacochiaceae, you get cyclic fatty acids. See hydrocarbic acid, saltmugric acid, gorlic acid, they are all typical of Flacochiaceae. Then all cruciferous plants oil will have erucic acid. Then all umbelliferae, they have petroselenic acid. Then we come to secondary metabolites. Now, these are the ones which help the plant to survive. And these are the defense mechanisms, defense or even offense mechanism. See, the plants also fight among themselves. So these are the compounds which help. They form the armory of the plant. And 3 major categories are there. There is alkaloids. Everybody knows alkaloid. There is no person who doesn't know what an alkaloid is. But how much they know, the true alkaloid we do not know. Then terpenoids. Few people know the term terpenoids. And phenolics, everybody says that they contain phenolics. But what are phenolics? Many people do not know. Then there are minor groups like amines, similar to amino acids. Polyacetylenes are there. Sulfur compounds are there. We will come to that. Then we come to alkaloids. Now more than 12,000, maybe now 13, because the number is increasing because of bioprospecting studies. You take any new plant that will contain an alkaloid. But alkaloids are bitter principles. See, if you go in the field for collection of plants, but that state is now not very common. Nobody want to go to the field and collect plants or study them. So Harboon says that if you want to know whether the plant contains alkaloid, just bite them. If the taste is bitter, that will contain an alkaloid. So they are bitter principle. As the name suggests, they are basic compound, alkaloids. That is the largest compound because people said that this is the largest. But now phenolics are the largest group. So more than 12,000, this is an old data. Now it may be 15,000. But the problem is that they are present in only 15% of the angiosperms. In lower plant, they are unheard of. They are not there. In algae, in fungi, they are present only in ergot or amanita. And in bryophyte, they are not there. Pteridophyte, they are not there. In gymnosperms, only Cephalotaxis, Ephedra, and Taxus contain them. In angiosperms, when the first angiosperm came, angiosperms, they attracted insects for pollination. So the insect they come to, because, angiosperm, what they did that they converted a branch to another flower. And the flower is having the same color of an insect. Flower is having the same fragrance. They contain pheromones. That is the brilliance of the plants. The flower contain in sex hormones of insects. So when an insect get the smell, the insect will think that my partner is there, then he will come to anything that is visible or that's the color one. It will visit the flower thinking that it's its counterpart and then go disappointed to another flower. So the insects are being cheated by the flowers, angiosperm. So then what happens that then they eat away. Because the ovules are there, then the carpels are there, they are all soft. So therefore herbivory started. So they are using it as herbal repellents. And they are present almost entirely in angiosperm. And four features of alkaloids. If you want anything to be called an alkaloid, there should be alkaline in nature. Then should be a plant product, even though some animals, sponges, they have alkaloid-like structures. There should be a heterocyclic nitrogen. You just see that. It should be a heterocyclic nitrogen. And then there should be distinct pharmacological activity. This is very important. There are many alkaline compounds are there, but not all of them are alkaloids. There are many products, they are not alkaloids. Many compounds are there having heterocyclic nitrogen. They are not alkaloid. But if all these characters are there, you can call them as alkaloids. Now I am showing some of the alkaloid containing plants. So you are familiar with what they contain. See ephedra. Ephedrine, you know that. Ephedrine is a group of alkaloidal amines. See, even though the title is given as indole alkaloid, that is alkaloidal amine. It is not a true alkaloid. It doesn't contain a heterocyclic nitrogen. But ephedrine is because of the pharmacological activity. It's an analgesic. For any type of fever, you use ephedrine. Then Rauwolfia, serpentina, reserpine. That is one of the first products accepted by allopathic practitioners for hypertension. Then Vinca, everybody knows that. Catharanthus roseus, Vincristin and Vinblastin. Now see that this is morning glory, hypomia. Now hypomia is very commonly cultivated as garden plants. This plant was very commonly cultivated in UK. And one time, many people came forward to collect the seeds. And then continuing year, there were no flowers. So then, the Scotland Yard, they started an investigation. They found that the people were consuming the seeds as hallucinogens. And, they contain ergot alkaloids. Ergot alkaloids is this one, ergotoxin. It has got a molecule, lysergic acid. See that this is lysergic acid molecule. And this, LSD, lysergic acid diethylamide. And all ergot alkaloids contain this hallucinogenic property. And these are very common in convolvulation. And one of my students, she worked on convolvulation. We have screened about 40 seeds of convolvulation, 40 different types of convolvulation seeds, and found that each one of them contained ergot alkaloid. But my boss said, don't publish it, because then people will get incentive. They will say, let us collect these seeds and consume for hallucinogenic purposes. Now, these ergot alkaloids and indole alkaloids are very characteristic of apocynaceae, asclepidaceae, loganiaceae, rubiaceae, convolvulaceae. That particular group contain almost every plant family, every genus contain as indole alkaloids or ergot alkaloids. This is isoquinoline alkaloids, that poppy, morphine, codeine, and all that. And this third picture is cinchona. This is having not iso, this is quinoline alkaloid. This is a different group. Now, argemone. Argemone is the latex of which you are using as Jivadaya Netra Prabha, that is used for eye problem. And everybody knows opium, poppy, and all that. This is very characteristic to earlier families. See, all Menispermaceae, the first families, Ranunculataceae, Anonaceae, Menispermaceae, or Papaveraceae, Fumariaceae, all of them contain these compounds. They are intensely bitter, intensely toxic to insects. So they were the mechanisms adopted by the early plants to ward off the insects or other herbivores. So morphine and berberin, all of you know that. So all the members of these family, then steroidal alkaloids are there in Hollerina, Solanum. See, when we come to bioprospecting aspect, I will come to these compounds again. Then steroidal alkaloids are there in Kurchi, Hollerina, Solanum. All Solanum, even your potato, they contain steroidal alkaloid. Tropane alkaloids, that is there in the Thura, Mandrake, coca, that is not Theobroma coca, Erythrosalem coca, that's a hallucinogen. Then we come to the second major group, terpenoids, which are otherwise known as isoprenoids, because they are formed of C5 isopentane. This is isopentane, having a head and a tail. And these two, they will join by tail-to-tail linkage or head-to-tail linkage or head-to-head linkage. And all the terpenoids, all isoprenoids, or you can call them as isopentanoids, all of them, they are made up of these isoprene units, C5 isoprene unit. Now, all these compounds will have some carbon here and there jetting out. Some tails are there, all of them, they will have some tails coming out. That is an indication that it is a terpenoid. Now, you see the classification. How many terpenoids you know? Volatile oil, essential oils. No, essential oils, the name is given because essences were prepared from them. So they, being volatile, they are known as volatile oil. And these were used by the plants for, they are antimicrobial. First time they were used by conifers. Conifers, they produced volatile oil in large amount. They have large resin ducts with monoterpenoids, and turpentine, all of them, they are volatile oils. Then they were adopted by flowers in angiosperms. Now, volatile oil, they are C10 compounds. Volatile oils are 2 isoprene units, monoterpenoids, 2 isoprenes. Then there are volatile oils which are high boiling, containing 3 isoprene units, sesquiterpene. They are very common in composite. Then we have 2 monoterpenes combined to form diterpenoids, resins. Now, 4 isoprenes, there are a number of compounds. Your stereoside is a diterpenoid. The amarogent, the bitterest compound, is a diterpenoid. And your gibberellic acid is a diterpenoid. Then angiosperm resins are triterpenoids, C30 compounds. That means 6 isoprenes or 2 sesquiterpenes linked tail to tail. And they contain steroid supportings. And then there are tetraterpenoid, that means 4 monoterpenoids, C40, containing 8 isoprene. You know them, they are carotenoids. They were produced when the plant were synthesized. And then the last group is rubber and gutta. They are polyterpenoids. They have more than, 10, 12, 15, or even thousands, rubber and gutta. These are the terpenoids. Then we come to volatile oils, monoterpenoids. Now, families Rutaceae, Labiataceae, Umbelliferae, Myrtaceae, Coniferales, etc., they contain volatile oil. And it's a characteristic family of those, characteristic feature of those families. Rutaceae means, okay, you break the leaf and smell it. It is a Rutaceae. Labiataceae, you smell the leaf, you will find that. Umbelliferae, you smell the leaf. Myrtaceae, conifers. So that is the characteristic feature of these families. Then another group, see, you know that Vitex or Lendana, the botanist, you know that if you make a herbarium, the leaves become black. The herbarium will appear black. It is because of these iridoids. Iridoids are first obtained from iris, an ant. That is why. And these are one of the most stable antimicrobial compounds. They are bitter also. But some of them are excellent medicines. Vitex is one of the best medicine for any type of rheumatism. Morinda, even noni. Then lendana is antiviral because of this compound. So iridoids, more than 650 in number, common in advanced families. See, they are non-toxic to the plant. See, alkaloids are toxic to the plant because they combine with the organic acids. And so the metabolism will be disturbed if there are more alkaloids. While iridoids are harmless, you can store them and you can use them. They are very common in Apocynaceae, Asclepiadaceae, Verbenaceae, Rubiaceae, Scrofulariaceae. You get loganin, you get dipetolactone. Diterpenoid, you get Astraceae, fever, echinacea. Echinacea is one of the best medicine for fever. And then diterpenes, all conifer resins contain, abitic acid, pymaric acid, all of them, they are rich in diterpenoids. Triterpenoids, there are many different groups. At least 8 or 9 groups are there. And if you refer any of my books, you will see the classification of triterpenoids. There are 8 different types of triterpenoids. But I wanted to say, what are the useful ones? Ecdysteroids. Ecdysteroids are insect molting hormones. And they were adopted by some of the ferns. So the insect eating them, their reproductive cycle will be disturbed. So all those herbivorous insects or any other insect which eat them, their larva will not be converted to glucose. So there is a very intelligent mechanism by those plants. And that is present in the whole of Caryophyllae. Caryophyllae means all your centrospermae. And if you remember Bandhamanukar classification, all your Amaranthaceae, Vagara, Amaranthaceae, Portulaceae, all of them they contain. See, this is Rajgiro from Amaranthus. whereas the seed, this is ecdysterone is one of the anabolic steroids. See, if you are a sports person, you cannot take any steroidal thing, but you can take ecdysteroids, or take only Rajgiro, or there are 100 other plants. Take Portulaca, and your body will be becoming healthy and fat. They are general tonic. Like that, in this, you take them as vegetables or seeds or anything, and that will be good for your body, good for your health. Now, there is another triterpenoids. Now, you know that neem and quassia. You know, quassia is Saimaruba, that is known as Lakshmi Taru. Now, neem is known for the bitter principle, and they are called lemanoids. Lemanoids because they were first detected from lemon. You know, the lemon juice, there is some disturbance. See, lemanoids are the light bitter compound present in a lemon juice. Now they are very good for the body. That is why they are lemanoids, and quasinoids. And they are triterpenoids. You know, azadirachtin, one of the costliest secondary metabolite, quasin. And all these are best biopesticides. And they are very good in fever, for resistance, antimicrobial. Then saponins, very common in sapindales and many other groups. Cardiac glycosides. They were used as arrow poisons because they have a direct effect on your heart. And you know, these natives, they used to take the latex of these plants and put at the tip of their arrow, and the arrow pierces your heart and you are dead. And then people then analyze. They say that in large doses, this will initiate collapse of the heart. But in smaller doses, if they will be used as cardiotonics. If the heart stops beating, give a little bit of cardiac electrode, it starts beeping. So many of them are used as medicines. Then carotenoids. See, carotenoids are the first examples where chemical characters are used in classification. All the algae are classified based on their carotenoids. See, you get red algae, you get brown algae, you get cyanophysic. And then there is rubberangata. Most of the lativorous plants like Euphorbiaceae, Moraceae, Astraceae contain. Then we come to next group, that is the phenolics. Sorry. Phenolics also, as a group, they were much despised. People said, yeah, what are the phenolics? They are hopeless. They thought only alkaloids and steroids are the only major compounds. But phenolics are the best antioxidants which help the plant in a highly oxidized environment. You know that now the world is changing, it is warming up. And the world around us is highly oxidative. So if you want to reduce the oxidation of your lipids or your tissues, you need large amount of antioxidant. And all the phenolics are excellent antioxidant. And you see in Ayurvedic, there are rasayana drugs are there, bodybuilding, all of them contain phenolics. And large amount of, there is no plant without phenolics. Now, you know, chilies, they contain capsaicin that is used as a pain reliever in Volini, rheumatism, curcuma. Now, they say curcuma is a medicine for everything. Ginger, excellent for rheumatism and for heart. Now, flavonoids. Now, flavonoids are yellow pigments. They are present in leaves. In leaf, they protect the chlorophyll from UV light. And all the white flowers which are blooming in the night, they are white because of flavonoids. And because of the flavonoid, they appear white in the night. And that is where the insect can see them. Now, flavonoids are present, but then you get at least 13 types of flavonoids. See, flavones, flavonoids, then catechins, tan, all of them are there. They are flower and leaf pigments, antioxidants, antimicrobials. Then flavonoids having hydroxylated, more hydroxyl group, rather hydroxyl group in the third position. Now they are very common in primitive group of plants. Now they are the best antioxidants. Now, three deoxy compounds are the flavones. They are characteristic to highly advanced group. Highly methoxylated flavones are still advanced. Then biflavones, dimeric flavonoids. This is typical of conifers. And some primitive woody angiosperms also contain the isoflavones. Isoflavones are isomeric to flavone. And this phenyl group is attached to third position. In normal flavone, it is at second position. This oxygen is the number one. So this isoflavonoid, this is very common in leguminosae. It's a typical chemical character of leguminosae, but you know, genistein and other isoflavones are excellent medicines. Then tannins. Tannins, because of their astringency, that's a defensive mechanism for plant. That is why a lot of tannins are there in the bark. Because the bark is exposed to the atmosphere. So you get catechin there. And then there are a number of other compounds are there. Then we come to sulfur-containing compound. This is very typical of Brassicaceae or Rhodales and related group. Your moringa, they contain the best antioxidants. See the antioxidants of Brassicaceae, glucosinolates. They remain in your body for about 4 days, and they will encourage all other antioxidants. They will make, and actually these are glucoraphan and all that, isocyanates are both antioxidants. They will make all other antioxidants work, and they will control. So if you take sulfur-containing compound, that is of Brassicaceae, that is why people say, you take more of moringa, you take more of cruciferous vegetables, that will give you immunity. It is because of their antioxidant properties. Then annonaceous acetogenins. See, this is the structure. They are hydrocarbons having 2 furan ring and all that. Now they are THF acetogenin. These are one of the best biopesticides. That is why they are present in Annona, all the species. Annona, muricata. Muricata is one of the best medicine for leukemia. And many people, you know, they take, you know, what do you call Lakshman Phal or Sida Phal or whatever it is. Annona muricata, graviola. This is said to be one of the best medicine for cancer. So people boil the leaves and drink it. But you know that people do not know that acetogenin decomposes at 40 degrees centigrade. So you boil it and the acetogenin is destroyed. And you drink it. And if ever you get a result, that is placebo effect. This is one of the best anti-cytotoxic, anti-tumor, anti-parasitic, pesticide and immunosuppressive activities. And this is an example. So I'm coming to end of the chemotaxonomical work. This is how chemotaxonomy can be benefits. And now, what is the potential of further research? And this is where every botanist has to look into. If Vinca rosea is not available, there are many other Vincas. There are many other species which are closer to Vinca rosea in the same Apocynaceae. The Asclepidaceae plant, they contain these compounds. Now, in chemotaxonomical work, you can see one of the best book is R. Tronquist's book, Integrated System of Classification of Flowering Plants. You may not get it, that is out of print. Dahlgren, another revised system of classification. Hegner, that is 73, and there are 16 books are there. That is in German. Then my one book is there, that is Taxonomy Evolution at Work. There also, I have followed these people and prepared my own modified classification. Then we come to bioprospecting. Bioprospect means searching a compound or searching a character in biologically related plants. That means if one plant in Scrophulariaceae is medicinally important, if it is not available, then you can search other members of Scrophulariaceae for these compounds. That is literally prospects of finding another source of beneficial compounds, beneficial characters among the plants of taxonomy group with the same external features, genetic makeup, and metabolic pathways. And this is, a substitute or an alternative can be sought in the near relatives of the source plant. You know that now everybody sings tunes on medicinal plants. They say that is the panacea. There is there. Everybody is jumping into that without knowing what plants are there, what the plant contain, what are the compounds there. And for phytochemical, chemotaxonomical studies would develop. Now you just see, this is one of my first slides I gave it in a lecture in bioprospecting. See the number of angiosperms in in planet Earth is 260,000. See, the God created the whole world and gave to you and me, saying that, son, I have given a rich wealth of 260,000, or with algae and fungi, it will be about 3 lakh plants. But how many are we using? How many are the useful plants to us daily? 100, 200, or 500? What are we doing with this planet? What are we doing with the rest 2.5 lakhs of plants? There are plants we are using only 20 cereals. And you know how many members are there in Gabonese? There are about 8,000 members are there. All of them, they contain food material. That is why, and I just took a illustration. See, in Bible, it is said, go to the ant, O sluggard, consider her ways and be wise. See, when I used to walk from my quarters to college, there was a big cricket ground. I see there were a lot of grasses and lot of ants going in line carrying the grains of those common grasses. And I used to wonder, if the ants can eat these seeds, why can't we eat? And that is why then I remembered the words in Proverb in Bible. It is written, go to the ant, O sluggard, consider her ways and be wise. Without having any chief, officer, or ruler, she prepares her bread in summer and gathers her food in harvest. Now see, we have a lot of food problems. We do not have grains, and there are 9,000 plants. And how many pulses we have? See how many dals we have. We have only 4 or 5. But the members of Fabaceae, legumes, 12,000 plus Caesalpiniaceae 2,200 plus Mimosaceae 3,000. That means 17,000 plants are available with beautiful protein-rich seeds. And what are we doing with it? So if you happen to meet God who created the whole thing, he will ask, my son, I gave you such a rich bounty of plants, resources. What are you doing with that? You are a very, very poorest manager of the planet Earth. This is my book. Now, bioprospecting of active and simple can be for herbal or Ayurvedic medicine, biopesticide, natural dyes, biofuel, biofertilizers. Now, this is my book on herbal technology, which deals with all this. Now, we come to bioprospecting of alkaloids, benzyl isoquinolines, that is morphine, berberine. Berberine is there in Daru Haldi, morphine is there in Papaver, the best pain relievers. Now, where you can expect them? Isoquinoline alkaloids. See, this isoquinoline, a group of them are benzyl isoquinolines. And see, important alkaloid isoquinoline containing plants are Berberis, Solangium, Tinospora, Poppy, Teliacora, and there are about 16 biopesticides are there. Raw nails contain 3,200 plants. Each one of them contain your precious benzyl isoquinolines. Papaverales, there are about 600 plants. So we have about 3,800 plants which contain berberin, alangin, morphine, codeine, etc. But we are just happy with what we are having. Now, Ranel, see, this is the breakup. Then ergotalkaloids. Ergotalkaloids are one of the best medicine. Ergotoxin is having that oxytocin effect, and it is very good for many other problems, migraine and all that. Then that is there in convolvulus, and then indole alkaloids like reserpine, vincristine. They are present in rubiaceae, apocynaceae, and asclepiadaceae. See, one example is the Rauwolfia tetraphylla. See, the Rauwolfia serpentina, that is your Sarpagandha contained reserpine. So, our Indian mentality, when Rauwolfia, serpentina was in short supply, somebody from Europe, they asked for Rauwolfia serpentina roots. Some of the fellows, our Indian fellows, that they said, what do they know about this? Let us put the Rauwolfia tetraphylla roots, which are morphologically very similar. And they could find out the difference. They say that this is less toxic than Rauwolfia serpentina. And it has the same activity, but without side effect. Now, your Vincar osea was discovered like this. Some people, they exported Vincar osea roots in place of Rauwolfia tetraphylla, saying that, sir, this is Rauwolfia, genuine Rauwolfia serpentina, Sarpagandha roots. Then they identified that this is not genuine. So then they gave a reply. You don't give me the genuine Rauwolfia serpentina. You send us the plant which you adulterated. We want only the adulterant. And that is how Vincar osea was discovered. Even so, even our dishonesty is also having some beneficial effect. Now you see how many plants you have. Apocyne is a 2,000 species. Asclepiadaceae is a 2,000. Rubiaceae is a 6,500. Logania is a 500 plants. See how many are there? About 10,000 plants are there with indole alkaloids. Terpenoids. We come to isoprenoids. See neem and cymaruba. Limonoids and quasinoids. One of the best biopesticide, anti-cancer compound, analgesic compound, and lot many good things are there. Now, Rutaceae, Meliaceae, Cymarubaceae, and Neoraceae, they contain about 2,000 plants. Only 15 plants have been studied. See, we talk about big limonoids and quasinoids and anti-cancer properties. And how many plants you have studied? 15. How many plants are remaining? 1,985 plants are remaining. Who will do it? You or me? Quasinoids, similar one. There are 110, one of the plants is in Kerala, they use it for cancer. That is called Lakshmi Taru. That is Cymaruba glauca. They contain quasin. And there are 110 different plants like Cymaruba glauca, cardiac glycosides. And these are present, and they are now not very commonly used in medicine because of toxicity, but they are now used in cancer researches. See, I have written an article in Facebook about cardiac glycoside. Latest research is that they are very useful in certain types of cancer compound. Where do we look into? For us, calotropis is a toxic plant. But for me, I give it to one-time remedy for all type of malarias. The same plant, a preparation based on the latex of that one. And you take that preparation, you will never get malaria, even seri malaria, in your lifetime. And this is the potential of plants. Cardiac glycosides, apocyne is a 2,000 species, asclepiadaceae is a 2,000 species, rubiaceae is 6,500 species, saponins. And all your medicinal plants like ginseng, bacopa, brahmi, licorice, then sedilla, curculigo, safed musli, kali musli, shadaveri, all of them contain saponin, biopesticide. 22 plants are there. all the members of Sapindaceae and others related families contain them. Ecdysterone, I have already said, there are 11,000 plants of Caryophyllaceae which contain ecdysterone. We can use them for our benefit, for having stamina, our resistance. Sesquiterpene alkaloids, that Jyotishmati, that is Celestris paniculata, one of the best herbal medicine for memory. I used to tell my people, China has only one jingo. With one jingo plant, China conquered the whole world as memory enhancer. We have 15 different plants, 2 types of Brahmis, 4 types of Shankapushpis, then lot many like this Celestris, maintenance, all those things. We have 15 plants still. We are not doing anything. We are never in the international market, not even 0.1% contribution we have. While we have 800 plants containing maintenance in or paniculative Celestris compounds. This is Celestris paniculata. Then tropyl and steroidal alkaloids, pain relievers like atropine, hyoscyamin, scopolamine, steroidal alkaloids, and Solanacea, about 2,800 species are there. Acetogenins, I told you only very few plants, 4 or 5 plants have been studied, but 2,300 plants are now there. Glucosinolates, one of the best antioxidants. So Brassicacea, 3,250. Caparidaceae, 625. Then sulfides like garlic. Garlic is a medicine for everything. Your heart, your rheumatism. And there are potential sources of sulfides are about 5,000. But how do we know about them? Unless you identify the plant, see, the first thing you have to do is identify plant. People do not know taxonomy. They say that, if you want to identify plants, you take the plant and go to computer. Uska gene sequence karo. Gene sequence to do. When are you going to do it? In the field, if you get a plant, if you cannot identify that, you are a dumb man there. So taxonomy is to be taught. Phytochemistry is to be taught. And then the whole scenery of the medicines and general tonics will change. Now mustard, these are. Then isoflavonoids. Genistin is one of the medicine for breast cancer. Rotinoids, biopesticide. Then a member of Fabales, that is leguminosae, 17,000 sources are there. And Genistin, this rotenone. Then we come to soybean, Pagamia, pesticide, lignans and coumarins. And you take flax seeds, you take chia seeds, all of them, they contain lignans and coumarins. But we have so many plants. Our tilli is much, much better than those flax seeds and all that. Now, so many plants are available. See, Linaceae, Rutaceae, Verbanaceae, Pedalaceae, Myrtaceae, Apiaceae. Now, this is flaxseed, volatile oil. And you know that is there in cosmeceuticals also. You can use medicines. You can use it as for fever and all that. Biopesticide, aromatherapy is there. Say, volatile oils are there in Rutaceae, Myrtaceae, Apiaceae, Astraceae. About 30,000 or 35,000 plants are there, volatile oil. Diterpenes. This is taxus. Iridoids, so many plants are there. Sesquiterpene, so many medicinal plants are there. Fever view, elephantopas, anti-cancer compounds are there. And they are present in all composite, there is Astraceae plants. Artemisia, one-time medicine for malaria, artemisinin is a sesquiterpene. Quinones, antioxidant. They are also best as natural dyes. This can be a dye for your food, dye for your medicine also. So all the carcinogenic natural dyes can be removed. Herbal cosmetics, you can use quinones. They are UV screens. They have better antioxidant activity. Cassia angustifolia, I have said that it can disrupt the protein envelope of your coronavirus. These many families are available with quinones. then phenolics, flavonoid, tannins, innumerable number of plants are there. Then tannins and dyes. Then these are some of the other examples. So if you get a new plant, see, suppose you get a new plant, how to proceed with bioprospecting? See, if you get a new plant or somebody brings a new plant to you, you find out the family, you go to a taxonomist if he is available somewhere around. But now all are molecular taxonomy, so they do not know the plant name. They do not know the family. They only know the genes. Now, if you can find out the family, then search the literature. What are the typical compounds of that family? And then search for active compounds in related plants. Then you take the extract of that related plant and co-chromatograph with this plant. And if it contains the same compound, see, you should know a little bit of phytochemistry, and you can have bioassay directed approach also. Then chemoprospecting, that is looking for the same molecule in a different group of plants. Then see, this is my book, so I should not recommend it, but this is the only best book that is available. Analytical Methods for Medicinal Plants and Economic Botany. More than 200 plants have been analyzed for different types of compound. This is written by me and my son, who is an organic chemistry, is a assistant professor in Navarajna University, Denny Maman. And this is published in 2016. It is sold out. Now they are coming out with a revised one. It costs 355. Then if you can afford, you go for plant drug analysis by Wagner, Blatt, and Skansky. It's a German book, but it is translated. You get absolute photographs of your TLC. See, color photographs. And see, this is right side, this is in UV light. This is UV in higher wavelength, 365. And this is visibly, and this also is UV light. So you can identify some of the compounds. And this is, I'm using this because one of my paper chromatograms, because I got material which is supposed to be Pterocarpus marsupium. That is Vijay sir, one of the best medicine for diabetes. So my manufacturer said, sir, I have a doubt about this. So I took the extract, just the methanol extract of that. That is this one with the standard plant I have. And you see, and then I saw it in UV. Absolutely the same. Quality control. Very, I said that you go ahead, this is the same thing. And this is flavonoids, just paper chromatography. And you can see my website. See, I was a very, very sincere teacher. So when I got retired, because I was sincere, my students came to me and said, sir, you were talking so much about these things. So why can't we do something? So we started a pharmaceutical, which they called it as Dr. Daniel Labs. It is not that I called it. They wanted its name to be this. And we have now 15 products. You can look to my website. We always look for the original plant material. And whichever material we get, a root, stem, or leaf, we can analyze them by either, if it's a plant material, by microscopy, or if it is an extract, we don't go for extract. Or we will find out the chemical compounds in that, marker compounds. So you just visit my this website and you get motivated. There is so much of, see, the whole world is waiting for you to come up with good medicines, good tonic, good nutraceuticals. See, you are being fooled by a group of thieves. They just blast your brain saying that this is good, which is absolutely trash. You can come out very good, but at least your health will be taken care of. I am 72. I use my own medicines. And the people who retired with me, they became very old now. I do not say that I am not old, but I am metabolically or I am mentally, I am very young. And you can contact me in my Dr. Daniel Snap gmail.com, or you can visit my Facebook account of Mom and Daniel, because I give a lot of stories on plant chemicals and plant adaptations. And if in acute cases, don't try to call me for any silly thing and all that, because I have lot many people just calling, sir, how can I do it? How can I do it? I said, no, my phone number is there. You can note this down, and you can get the recording of this lecture. And if I could enthuse you a little bit, even by 5%, I know that my this effort will be successful. Thank you very much. And if there is any question, I will be. You can contact me in my email, or if Madam permits, you can ask me now.
Thank you, Dr. Daniel, for such an enlightening lecture. Now I request the participant, if you have any question, please do ask. Sir, can we have the question answer session now?
Yeah, sure. Go ahead.
So anyone having any question, please do ask.
I think all are happy.
Yeah.
I
Any
think that-
question, please? Okay. If there is no question, so I take this opportunity on behalf of the Department of Botany, RD National College, to thank Dr. Daniel for accepting our invitation and giving such an enlightening lecture. I thank all the participants for attending this session. I thank our principal for supporting all our activities, Dr. Milind Kulkarni for all the technical help, and our DBT Star College team for the support. Thank you so much.
Thank you, Suchindra ma'am.
Thank you.
Thank you, Daniel sir. Can I end?
Yes, sir. Yes.
Oh, yes. Thank you so much, sir. Thank you.
Thank you.
Okay,
Thank you,
fine.
Milind. Thank you, Milind. Thank you,
sir.
sir.
Thank you, ma'am.
Milind, we can end it now.
Yes.
Yes.
I'm